Welcome, everybody, to another Purdue Lecture Hall series. My name is Tommy Soares. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And today I have the pleasure of welcoming Dr. Eric Kontowicz to our Lecture Hall series. Thank you, Eric, so much for doing this. You come highly, highly recommended by Professor Wendy Boves here in the Comparative Pathobiology Department in Veterinary Medicine here at Purdue University. And Dr. Boves focuses on the transmission of infectious diseases in animals and in nature and how it happens from species to species. And you're going to talk to us a little bit about your research, but I know that everybody here is curious to hear not only about what you're doing now and how that research works, but how you got here and how you started doing this work and all of that. So welcome to the program, and please, I'm gonna let you take it away, and I'll be quiet now.
Well, thank you. Thank you very much for that awesome introduction. Hopefully I can live up to it. So yeah, I'm going to talk to you mainly about our work in Dr. Boves' research group. And this is a really fancy title that basically boils down to, we're looking how flu can be transmitted between hogs and people. I guess to give a little background on myself, because I don't have slides on this, but I started, I guess, my research career more or less at Michigan State University. It's where I got my undergrad degrees from. And I kind of started just, I don't know, I bounced around to a lot of different places, doing a lot of different things, but infectious diseases always kind of interested me the most. And then I went on to get my master's in public health at Michigan State as well, where I looked at some sexually transmitted infection data and did some research there. Because again, I think kind of infectious diseases and that intersection of public health and behaviors is extremely fascinating. And then at the University of Iowa is where I got my PhD in epidemiology, and that's really where my love for zoonotic diseases came from. And zoonotic is just, again, another fancy name for diseases that could be transmitted from animals to people and sometimes back again. So flu is a perfect example of a zoonotic disease. I did some aspects of my PhD dissertation research with flu. And that's kind of what brought me over here to Purdue, where I'm doing my postdoc now. So that's a very long-winded way of saying I've had kind of a windy, curvy path to getting here, but for the most part, it's all research, and sometimes that's what research is. So yeah, let's jump into this fun research that I have here. So to give a little bit of a background of, this research in general, we know what flu is. It's a member of the Orthomyxoviridae family of viruses. It's very common. I can guarantee that every single person probably listening to this has had flu or knows someone who has had flu. It's exceptionally common, but it goes common to both humans and animals. I like to give the antidote of, if you have lungs and you breathe, you can get flu, because this can even infect whales, seals, birds. But in regards to this presentation, we're mainly going to be focusing on people and pigs. And typically it's transmitted via respiratory droplets, definitely in pigs and people most predominantly. And we know domestic pigs can play a role in the maintenance of influenza worldwide. And it's because pig production is huge worldwide. There's a lot of global transportation and trade. And then another reason I don't have listed here is pigs can actually be infected by almost every strain and subtype of influenza out there, regardless of where it's coming from. Most importantly, pigs can be infected by flu that are origins in other pigs, origins in avian species, and origins in human species. And we'll talk about that in a little bit too. So what's it like on a hog farm? Well, influenza poses a really consistent threat for the hog industry. A single introduction of flu can really rapidly spread throughout all of the hogs or the pigs on that farm. And that's a lot, mainly due to the high density of animals, provides the optimal conditions for spread. So you can't really social distance or quarantine like we heard during the COVID-19 pandemic. It's hard to do when you have 4,000 pigs kind of densely compacted into these barns. We also know that interspecies transmission can occur, and interspecies just means between species. So it can go from workers to pigs. It can even go from pigs to workers. And we've seen swine workers have elevated odds of antibodies, which just means they have a higher likelihood of exposure to influenza from swine or swine origin influenza. And then we've also seen, or it's been evidence that swine workers can consistently reintroduce 2009 H1N1 back into pigs. So this was really prominent during the 2009 H1N1 pandemic. And conditions like this can really help give rise to a pandemic subtype of influenza. So we've seen this in 1918, we've seen it in 2009, and then there's also many other pandemics that I haven't even listed here because they would take up way too much room on the slide. So we also know that influenza can pose economic and health risks on a swine farm. In particular, influenza can become endemic within these hog populations. So it's got that respiratory transmissions, like I've already mentioned, and you have densely populated conditions. We also have new piglets coming onto these farms about every 20 to 26 weeks. So an outbreak might be dying off, and then all of a sudden you introduce now a whole new set of susceptible pigs, and now the outbreak can perpetuate. And once a pig is infected with flu, it can actually affect their growth. So they can lose about 5 to 12 pounds, and it can prolong them reaching finishing weight or marketing weight. And these prolonged finishing times can really place an economic burden on our farm owners because now they have to feed pigs longer. It takes them longer to reach that weight before they can, return on their investment, essentially. We also know that mortality can play a factor. It does vary from subtype to subtype. So some, flu strains that are out there are worse than others, but generally speaking, it's around 1%. But again, if we're thinking about 4,000 hogs, 1% isn't a nothing number. It's still a good number of hogs that are being infected and potentially dying. However, in naive populations or certain subtypes of influenza, we can even see that mortality rate go a lot higher, up to like 10%. So now if you're on a farm of 4,000 hogs, that's 400 hogs that have passed. And you can also have public's perception of influenza. In fact, we saw in 2009, pork consumption in the United States dropped almost to an all-time low because people were afraid they were going to get influenza from the pork meat. That's not going to happen. You can't do that, especially if you're cooking your pork meat, but that's not how that works. But so there's a lot of factors that can even drive economic burdens of this disease. And then we also have when workers have sick days, that can greatly impact the daily routines, given the low numbers of workers that are needed on these growing or finishing farms. So to give you an example of what we're talking about when we say densely packed, this is one room on a pig farm. And there's actually 4 of these, especially for the models that I'm going to run and show you today. But you can see these pigs are densely packed. They're essentially on top of one another. It's really easy for a respiratory agent to just run wild through all this. And then you can imagine that any worker going through here too, or interacting with these pigs, might be put at risk at breathing the same air and potentially getting infected with influenza here. And then they can leave this door, go right to the other side, and there's a whole nother room that looks just like this and potentially spread or transmit the virus to all of those pigs. So how do we stop influenza? Well, there's a variety of control measures. Most common, we see vaccination. There's a number of different strategies. You can vaccinate the pregnant mothers or the sows, and that in turn imparts maternal antibodies onto the piglets, or you can even vaccinate piglets after they're born and like before they're sent off to the finishing farms. In some places, they'll do both. Some places will choose to do one or the other, and other operations will choose to do neither. So there's really kind of a wide variety of what we see for vaccination practices. We can also change the way our workers operate. They could always work from our younger hogs to our older hogs. And I'll have a couple slides that explains this a little bit better, but we see this in our poultry operation where they'll always work from the youngest chicks to their oldest chicks. And what this does is really helps those younger pigs or chickens that have a more naive immune system, helps protect them from anything that might be circulating in the older animal population. We can also improve our equipment cleaning, our airflow, and our worker personal protective equipment, or PPE. This can really help reduce the likelihood of indirect transmission, or what we're calling between room transmission. So as workers go from room to room to room, influenza can actually can get embedded in their clothes, in things that are called flow mites, or things on the equipment too. And that can go, if that's not cleaned properly or not sanitized properly, that just basically just serves as a different contamination source or transmission source for flu to other pigs. And then we can even quarantine our sick hogs, although this might have limited effects just because you don't have a whole lot of room to quarantine. There's some places that they can take a number of hogs out at a time, but you can't really quarantine 3,000 hogs or anything like that. It's really difficult. And then we could also increase our worker PPE, so just improve its quality or improve the adherence to it. So the objective of this study in particular is we wanted to look at those control measures and look at and how that impacts the number of pigs that are infected, the probability of our workers getting sick, and the timing of our workforce getting sick. So to do that, we built a metapopulation model. And this is really the main thing to take away from this is we see 4 rooms. So each of these rooms has 1,000 hogs in it, and they're filled at different times. So this is filled on like day 1 or day 0. And then 2 weeks later, this is filled. And then 2 weeks later, 2 weeks later. And every pig that comes in starts at 3 weeks of age. So 3 weeks of age, but 2 weeks later, they're now 5 weeks of age, where this is 3 weeks. So this is what I mean by this is also our older room, and this is our younger room. So each pig, we also ran a variety of scenarios, and each scenario had 1,000 iterations. So we essentially ran that same scenario 1,000 different times because we incorporated some randomness and we wanted to see what that did. And then on the last room being filled, that's where we introduced our single pig that was infected with flu and was able to transmit to other pigs. We assumed density dependent transmission because, again, a lot of these animals are densely packed into these rooms. And our pigs and workforce didn't have any acquired immunity as a result of like a previous infection. So we just assumed pure susceptible populations here, which is not necessarily the perfect representation of real world, but we wanted to kind of think about the worst case scenario. We also assumed homogeneous mixing of pigs and workforce, which again is a fancy way of saying our workers had an equal likelihood of interacting with every single pig on any given day. And none of our workers were considered to be vaccinated. So if you think about those 4 rooms, these compartments is what's happening essentially in each room. So this kind of signifies how a pig or a worker will move through the workforce or move through the disease process. So essentially a susceptible pig comes in contact with an infected pig and they become exposed at some contact rate. And then after they're exposed, they're not infectious yet. So they're pre-infectious for some latent period, and then they become infectious. And they remain infectious for an infectious period until they become recovered. And once they're recovered, they can't be infected again. And we incorporated a stochastic approach, which just means we used a little bit of randomness involved, because just because you come into contact with an infected person does not guarantee that you will become infected yourself. So there's a little bit of randomness involved in the disease process, and that's all that means right there. And we built off previous work done by these 2 research groups, where we really, the novel thing we're adding is our workers here and how they can become infected from pigs, and they themselves can actually serve as an infectious pool to infect pigs. So this is all of our model parameters. I know that there's a lot here, so don't worry, but I wanted to say, like, we built this using a lot of different literature. However, there is kind of a lack of really good literature and experimental data that's super controlled to gather all of this information from, but we were able to find some good experimental data looking at pig-to-pig transmission of pandemic H1N1, and that's how he developed this beta parameter. So how fast susceptibles can become exposed. And then as far as our workforce to our pig transmission, again, this was rough to find, but we did happen to find a paper looking also at pandemic H1N1 at a research farm in Alberta, Canada. So that's how we came up with the transmission parameters from workers to pigs. And then to come up with our pig to worker transmission, we found another paper. This unfortunately looked at H3N2, but I could not find anything that was looking at H1N1. And this was also at an agricultural fair. So outdoor events, but again, finding data was a little tough. I was truly controlled and experimental to look at pig to workers. But this is all the data that we use to really drive a lot of our model. So in particular, we look at a number of interventions. We looked at pig vaccination. And so when we say mass pig vaccination, that's every single hog receives the vaccine. And it was either 40% or 80% efficacious. And all this really corresponds to with the percent of the susceptible population that went directly to recovered. So if we had a 40% efficacious vaccine, it just meant 40% of our susceptible population was considered to not be susceptible to the virus anymore. So we just moved them directly to recovered. And same thing for 80. We also looked at quarantining of sick pigs. So in order to do that, we essentially created a separate compartment. So instead of just susceptible, exposed, infected, recovered, there's now a quarantine compartment. And for each room, this quarantine could only hold 10 pigs because we wanted to put a realistic approach on this. Again, these farms don't have unlimited space and unlimited time or unlimited capacity to just put pigs into quarantine. So essentially, if there was space, pigs were either moved to this quarantine or not the day or 2 days after they became infected. So it went into that I category. And this also, we also assumed here that workers worked with these quarantine pigs on they were the last group that they worked with. So there wasn't a chance of them spreading anything that was within this quarantine population to other populations of pigs. We also looked at changes to our workforce routine. So again, always working from our youngest pigs to our oldest pigs. And again, this can help shelter our younger pigs from anything that might be circulating in these older pigs. And then lastly, we looked at what happens if we improve our workers' personal protective equipment and their adherence to it. So we tested either them wearing a mask, or not wearing a mask, wearing like a surgical or a cloth mask, and then wearing an N95 respirator. And essentially, this just corresponds with a % reduction in the interspecies transmission. So if you're wearing a mask, that greatly reduces your chance of spreading it to another person or another pig. Whereas if you're wearing an N95, that also protects the wearer from picking anything up too. So to jump into our results a little bit here, we can see this is what happens on a farm that doesn't really have any of these control measures in place. So no vaccination, no worker routine, no quarantine, no nothing. So this is just our baseline model. And we can see here, our rooms are being filled. So on day 0 is our first room, day 14, day 28. So just 2 weeks apart, boom, boom, boom. So room 1, 2, 3, 4. And on this last room is where we incorporate our first infected pig. And we can see shortly after, our infected numbers start to rise. So our susceptible population drops. That's the blue line here. And we can see overall that over the course of this outbreak, almost every single hog is infected. All but, what is that, 42 of them? So pretty much every hog is infected over time. And we have a probability of our workforce infection being 0.61. So that's a little worse than a coin flip, if you want to think of it that way, because 50-50 would be like heads or tails. And it takes about 3 weeks for our workers to be infected. So what happens when we start to incorporate different control measures? So to orient you to these figures here, because you'll see something like this for the next couple slides, our control measures across the bottom. And then on this left graph will always be the probability of workforce infection from 0 to 1. And then on this right graph will be the days to first worker infection. And this is days since that infected pig is introduced. And here, this pink/red box is always going to be our baseline model. And these numbers are the number of total infected pigs for this scenario. So we can see as we improve our vaccine efficacy, it really drives down the probability of our workers becoming sick. And it also drives down the number of total infected pigs. Those things are most likely very correlated, right? And we can also see that as we reduce this probability of our workers being infected, generally, takes longer for our workers to become infected. So that's something that we want to see. So we can see if we improve our vaccine efficacy, for the most part, we can really drive down not only the total number of infected pigs, but how often or how likely it is that our workers are going to be sick. And 40% is about what you would expect to see in today's world, where we see about 40% vaccine efficacy. However, if we are able to make an 80% vaccine or a vaccine with 80% efficacy, we could see vast improvements. So something to strive for. So what happens when we quarantine? Again, if there's no quarantine, this is our baseline. However, if we are able to quarantine a pig 2 days of it becoming infectious, we still see a big number of pigs becoming sick, but we do drive down a little bit of our worker probability of becoming infected. However, if we are able to quarantine a pig within 1 day of it becoming infectious, we can see this drastically reduces the number of total infected pigs and does overall drive down the probability of workers being infected. And we can see this same general trend of it takes longer for our workers to be infected as well. If we change worker routine, so they're always working from our youngest pigs to our oldest pigs, and then we look at what happens when we introduce an infected pig into different rooms, we can see actually if the infected pig is introduced earlier or within our older pig population, can drastically reduce the total number of infected pigs and the probability of our workers being sick. However, if the sick pig is introduced, within our younger populations, it still pretty much results in a whole scale farm outbreak. But this is a nice thing that does correspond with, this is a very, for the most part, a simple, no cost change to something that we can do to help really quell an infection or influenza spread on a pig farm. Because if we do know that there are known pigs that are sick or rooms that have known sick pigs, we could potentially work with those pigs last to help drive down the likelihood of us transmitting influenza to another room or even in turn to our workers. And we don't see too much of a change as far as days to the first worker being sick, because again, this is calculated from the days of that first infected pig showing up on the farm. And we're not doing anything else. So they're not like wearing PPE. We're not vaccinating our pigs or anything like that. So I didn't expect to see too much of a change here. So what happens if we actually look at all of these things? So if we not only vaccinate our pigs, but we implement a quarantine system and we change the way our workers operate, we can see that we can drastically reduce the total number of infected pigs down here, almost all the way down to nothing. We're still going to see one because, you're going to have a pig that comes in that's going to be sick. But we can really also drastically drive down the probability of our workers getting sick. And then, as I said, 40% vaccine efficacy is about what we anticipate. However, if we can get an 80% efficacious vaccine, we can see that this almost completely reduces the likelihood of an outbreak, and it almost brings our probability of our workers being sick almost down to zero. So once again, something to strive for, and if we can incorporate all of these different control measures on a farm. So if we happen to improve our worker personal protective equipment and their adherence, so these are kind of results from what happens when I took all of the same parameters that I used in our baseline model, except now I just made a worker sick instead of our initial infected person or entity on this farm was now a worker and not a pig. So that's really all that's changing in this scenario. And I didn't have any vaccinations for the pigs or no other control measures. So this is really only looking at what happens if we change worker wearing PPE. So we can see here that the probability of a worker infecting a pig was again about 0.54, so just about a flip of a coin. And the total number of infected pigs, though, was still very high, almost the entire farm. So while it wasn't necessarily a super high likelihood that a sick worker could get a pig sick, once it did happen, we could see a huge number of pigs getting sick. So when a worker wore a mask, so this again is just like a surgical or a cloth mask, we could see that the probability of the pig being infected was reduced to 0.28. However, once it did reach that population, there was a good variety or variation here, but we could still see a whole wide scale outbreak across the whole farm. But for the most part, it didn't really happen as much. And then if a worker wore an N95 mask, the probability of a pig being infected was reduced all the way down to 0.1. But again, if it did happen, there was a chance that you'd have a whole wide scale outbreak across that entire farm. So in conclusion, we do know that our current control measures on a typical indoor hog farm are not sufficient to control the spread of influenza between pigs and to the workforce. We also know that vaccination alone can reduce the total number of infected pigs. It can also reduce the probability of our workers being infected. However, thinking of that industry standard, about 40% efficacious vaccine, we still see about 50% of our pigs being infected. So that's still a good chance that you're going to have prolonged finishing times, take some longer to reach that market weight. So you put economic burden on your farmers, and not to mention the mortality rates of these potential pigs. We also know that quarantining sick hogs may also be insufficient. We did see that if we were able to quarantine the same day that a pig was sick, that can have an impact. It can reduce the total number of infected pigs and the probability of our workers being infected. However, there's limited space to do all of these awesome quarantine measures on a real life pig farm, short of telling a farm owner to just build a whole new quarantine pen, which that's a big investment that might not happen. And this can also be further complicated by the idea of asymptomatic transmission. So what happens when a pig isn't showing outward symptoms, but can still transmit the virus? This is something we did not model in this current work, but we will be in the future. We also know that workers strictly adhering to routine of always going from those youngest pig batches to the oldest pig batches, that can really have a phenomenal impact on reducing the probability of workers being infected and the total number of infected pigs. So that's really helpful when a vaccination isn't present. So say there is a novel subtype of influenza that crops up and is running rampant across these pig populations. Implementing something that changes the worker routine might help drive the total number of infected pigs down. And this routine could also be implemented to help ensure that workers are always working with sick pigs last. So if they know that a room is currently experiencing influenza, they could work with that room last to help reduce the likelihood of spreading it to other pigs or even to themselves. And then lastly, we see that improvement in PPE and adherence to PPE mandates by workers can help reduce the probability of our workers infecting our pigs. So again, without other control measures in place, an outbreak still can occur, but this is still showing a positive impact at reducing the likelihood that a worker can get a pig sick. So again, despite all this work, we do know that there are some limitations. Our transmission parameters were based on limited data. There's a wide variation between influenza subtypes and different conditions that could impact the way influenza is transmitted. But again, there's kind of a lack of really awesome experimental data that has perfect control groups, perfect test groups, and all that. So we're just trying to use the data that was available. Our current model also only includes a single influenza virus with no cross protection and a single production cycle. So we basically stopped this model at the end of 26 weeks. It is likely that the total number of infected pigs or even the probability of workers maybe being reinfected could go up if we start to incorporate, all right, room one now emptied because they reached all those pigs reached market weight. So now we bring in a whole new batch of piglets, a whole new batch of susceptible populations. And this could really help this influenza outbreak just be a perpetual problem on the simulated farm. Also, if we incorporate different viruses, so different subtypes, what happens when, let's say you have H1N1 circulating, but then H3N2 begins to circulate, what might happen there? Our results from the study also may not apply to outdoor units or facilities that are not grower specific or anything that are not outside of the US. So again, we're very US centric for this model, an indoor model essentially. And then despite all of these limitations, we do think our findings demonstrate that relatively practical interventions could be applied to reduce transmission in order to mitigate the economic and public health impacts, especially in the face of a novel influenza subtype. So there's definitely some practical measures that we can take if a novel flu virus does happen to crop up to try to help mitigate the spread of that virus. So with that, I thank you all for your time, and I'd love to take any questions that you may have.
Thank you so much, Eric. So interesting that the whole thing, and I think you did a great job at trying to explain it to us mere mortals who didn't have to do the calculations and computations on the modeling. I mean, it seems like there is a lot of computational work that you do in this work. I mean, you have to know biology, you have to know epidemiology, you have to know statistics, you have to know computation. I mean, I mean, like, you have a lot of tools under your belt, as I hope the people that are watching this program and are seeing this can really tell us a little bit how did you pick up all of these tools that are on
Oh.
your belt now?
Over the years, this is definitely not something that just happens in 1 year, right? Although I do have to give a very big shout out and thank you to Dr. Wendy Bove, because she helped me navigate. This is actually the first time I've done a formal compartmental model. So if let's see if I get out of this slideshow and just go back up to the, where I showed the susceptible, exposed, infected, recovered. This is kind of the first time I've ever really done something like this, but building this tool set was really just an accumulation of all the different educational experiences I've had. I've been fortunate through undergrad and graduate school. I've taken a variety of courses that were like, oh, hey, we're going to work with this computer program like SAS or RStudio or anything like that. So it's really just been over the years. And then the PhD is for better and for worse, it gives you a lot of experience firsthand. And I think the best thing it gives you is a lot of problem solving. I learned how to Google a lot of questions, especially with errors in code, because you're going to run that. No chunk of statistical software code is ever going to run the first time perfectly, especially if it's your first time making it. So
Yes.
Trying to Google and learn. And then the other big skill that I learned was when and how to ask for help. So sometimes you can't quite understand what Google's telling you, or you still can't understand, but there's people out there either in your college, in your department, or across the country or across the globe, really, that can be helpful. So sometimes just sending that email going, I have no clue what to do, please help. And
Yep.
I have found that for the most part, people are willing to help.
So, Eric, when you go back and you think, oh, I really wanted to do infectious disease, was it a day that you found that out? Like, was it a particular revelation that you had that you said, oh, infectious diseases is what I want to do with my life, or that's what I want to study?
Yeah.
But
I'm trying to think. So I don't know. I mean, when I started undergrad, I was very much along the lines of, I'm going to go be a physician. I'm going to be a doctor that works in a hospital and does, like, I love the show Scrubs. It's still one of my like all-time favorite shows. So that's really kind of what drove me to medicine. And then I actually suffered an accident in my freshman year of undergraduate that rendered me paralyzed. So I'm in a wheelchair. And through that whole experience, I spent a lot of time in a hospital. I actually have a family member too who's a surgeon, or was, because he just retired. So through all of those years and all those different experiences, I was like, I've spent enough time in hospitals and doctors' offices. So I kind of transitioned out of wanting to be that. And then I took a class my last year of undergraduate, and that kind of opened my eyes to public health. And I've always been fascinated, I guess. What draws me the most to infectious diseases is it's a single virus or a bacteria that can have these huge and traumatic impacts on the global population. I mean, 1918 pandemic, or 1918 flu pandemic, right? That killed anywhere to 40 to 70 million people, I believe, depending who you ask and what textbooks you're reading. Talk about COVID-19 and how that completely upended a lot of society as we know it. That's a virus. That's not, like, it doesn't have a brain. It doesn't think. It just does what it's supposed to do. So I've always found it, I guess, laughable how we think we're the top of the food chain, but we can be taken down by a single-celled organism that can't think and just completely take you out. So that's really where my fascination came from. And then it just over it, like I like history and like reading about all these different plagues and pandemics and the way we talk about different viruses or bacteria or just any of the different diseases that have threatened human population over time. It's fascinating how much we've learned and haven't learned over the years. So that's really kind of where the whole fascination kind of came from and just what stuck with. And then I went into epidemiology finally because my bachelor degrees are in physiology and human biology, and then I have my master's in public health. So epidemiology felt like the perfect marriage of those two things where I can bring a lot of the cellular ideas that I learned through physio and human bio and a lot of the population level science from public health. So that was a very long-winded answer, I think, to your question, but hopefully it
I
helped.
think that's perfect. No, it's perfect because it lets us see a little bit, gives us a bit of a window into your journey and your process there. Did you do a lot of research in your undergrad then?
No, not too much. So I was very fortunate. I think it was my sophomore year where I did in my bio 2 class, I was I had a phenomenal professor that like completely changed. Because at first I was like, ew, I don't want to do anything research. That doesn't sound like fun. I got to write papers and stuff like that. No, thank you. But he kind of opened my eyes to research, and it was because he had a very student-driven course. So he kind of set up the parameters of it has to be a genetic disease and you're doing PCR. Like that was all that it was. And then it was like you pick the genetic disease with your group. You actually had to design the primers. You had to order the primers. So getting like hands in the science in a classroom setting was really nice. And then I took another class with him a couple of years later, and that was all cystic fibrosis related, so not infectious at all. So that's kind of my extent of my research in undergrad. So I did not do a lot. Most of my research experience really came from kind of the last year of my master's, and then all through my dissertation work or my doctorate work in Iowa, where I did a bunch of different. So I had my own dissertation that focused on flu and Lyme disease and developing different models to predict those two things. But the lab group that I was a part of, we actually looked at Leishmania, which is a parasitic disease for dogs that can also be transmitted to people. Not so much in the US, but we do see issues in other parts of the world.
Yeah, and incredible, as you said before, how so many of these single-celled organisms or even viruses, if you don't want to call them single-celled, right? Yeah, that are so dangerous and they cause so much havoc.
Yeah, I mean, for me, it's like a flu virus and coronavirus. They're cousins because they're both single-stranded RNA viruses. That's it. So it's a single strand of RNA and some proteins that they make that really can shut a whole world down. And it's fascinating to me because it's the easiest thing to do. I could say to like, okay, this is how we prevent it. Just don't breathe, right? Because that's the only way to really stop it. But obviously, breathing is essential for life, so you can't do that. So now it's like, okay, what do we do with our behaviors to help minimize that spread? So
I really like that because there's a lot of, as you said, there's a lot of procedural things that we could do to improve the efficiency of an operation like this in the farm where you're dealing with so many animals. And it just seems almost like, oh, this is a no-brainer. Like, oh, why haven't we done this before? And you realize that maybe sometimes we didn't have the tools. We didn't have the Dr. Kontowicz's there to help us create the tools on the now able computers that we could do it on. Tell us a little bit. I mean, like you've said a few terms here that I hope everybody watching by now understands. Okay, if I don't know what epidemiology means, I hope that you're going to go check it out. Or, you said PCR. I hope by now most people, after going through the pandemic, will know what PCR is. But take us back a little bit as to, these tools that you're creating for people to be able to improve the operations in the farm, which is, people think that, Dr. Kontowicz is only interested in transmission of the infection. And you would be like, yes, 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 but why?
Yeah.
What's the purpose? And that is so awesome to hear. Tell us a little bit, I mean, again, historically, and why we can do some of the things that we're able to do now.
I mean, yeah, so as technology progresses and anything like that, right, we develop more tools, or that's, to me, the most fascinating or the most awesome thing about science is the drumbeat always keeps going. We might mess up at some point, but for the most part, over time, we'll kind of correct that mistake. So we'll think, heck, to go back to that 1918 influenza pandemic, we didn't even know what viruses were in 1918. We thought it was caused by a bacterium that actually has now been called Haemophilus influenzae, which is a bacteria that co-infects individuals with influenza virus, because we couldn't culture viruses way back then. It was actually a researcher in was it the 40s or the 50s that went up to some glaciers in Alaska and found individuals that had passed during that pandemic, resumed the bodies, took samples, and was like, oh, look, we have a virus. And that's where we were able to find the 1918 pandemic virus. But yeah, it's just as technology progresses, you learn new things and you learn how to do things, or more importantly, you can do things faster. I mean, PCR used to take 24, 48 hours. Now it can take 30 minutes and you'll have a result. So it's just kind of the fascination of technology, but I think you bring up a really good point. Never lose that why. So, we know that flu is a big problem, not just for humans, but for all these different animals across the world. And you can't save every animal. You can't save every domestic animal. You can't save every wild animal, but anything that we can do to try to help minimize that burden. And a virus, to quote Jeff Goldblum from Jurassic Park, life finds a way. Like, a virus is going to find a way to get around what we do. But if we can build up some actual barriers to just mitigate, if you put on that respirator or put on that mask, that's a cloth barrier that a virus can't mutate around. And it's so trying to think about those things of like, all right, what can we do to help protect our farms and our farmers, but not put them under more economic burden? Because yes, let's say we come up with this awesome vaccine, but if it costs them $1,000 for each pig, they're not gonna do that. So it's like, all right, what can we do that's like almost no cost, but they can still have a positive impact at mitigating this virus transmission?
I think that's exactly right. And because we're always thinking, oh, we just have to throw a lot of money at this to solve this problem. And that's not always the case. And as you've shown with your work, we can improve processes, we can improve placements, we can improve the way the workers interact with the animals and the way that we situate them. I don't know if you know the Nipah virus story, but it's a classic one where, the Nipah virus was being transmitted from bats to pigs because there were these fruit bats that were eating mangoes at night and dropping saliva laden pieces of mango that then the pigs that were being raised underneath the mango tree were eating, and they were being infected and then infecting the farmer. And then the solution was just move the pigs away from the mango tree. And this is exactly the beauty of your work. It's like, wait, there's a process here, and the solution might be much more inexpensive and much more procedural to improve the efficiency of way we're moving things. So I appreciate that, your experience throughout has been a building process. Tell us a little bit. Were you ever scared that you were doing the wrong thing or that you were going down the wrong path? I mean, that
Oh
because I
yeah.
imagine many of us coming from high school to college, now they're making us decide what we want to do for the rest of our lives.
Yeah.
And, like it's a scary thing. So take us back if you can.
Oh yeah, that's no problem. Man, so as I mentioned, when I started college, I wanted to be a physician, and that was something that was kind of drilled into my head from a younger age of like, oh, if you're like job security, go be a doctor. No robot's going to take over being a doctor, or anything like that. So that was kind of, and I was just more or less fascinated with the human body and medicine, and then Scrubs, the show. So like that's really where it kind of all started. But I don't know. I've actually given this tidbit of info a lot. Take your undergrad and even part of your graduate experience, if you continue into that course, to learn what you don't like and what you don't want to do. Because I think that's more powerful than finding like the one thing that you just fall in love with. Because I mean, I'm not going to lie, it changes sometimes on a day-to-day basis for me.
Yeah.
Some days I love sitting behind my computer and typing up lines and lines of code and trying to figure out what the problem is. And the next day I'll hate it. So that ebbs and flows. And yeah, there's a lot of days, especially through grad school, where you question yourself of, did I make the right choice? Did I do the right thing? Not only just with like life choices, but even in your research of like, is this really the right thing? You get that one reviewer that reads your paper and just calls your entire like life's work at that point garbage. And why'd you even submit it and think this was worth it? And you're like, well, cool. So
Yeah.
it's, you kind of got to keep going. It's to, night's darkest before the dawn type of deal. But really learning what you don't want to do can be just as powerful, if not more so, than finding like that one thing that you think you love. Because I also have friends that have gone through programs and then turned out they hated it. And, they went through med school and they're like, I can't do it. I don't like it. This sucks. And they have to pivot. And that happens too. And that's fine. It's all about the journey, not the destination. And there's no direct path. Having curves and turns and detours and trying to figure it out is also fine. As long as you're trying to learn something along the way, I think that's what's the most important. So yeah, again, I think a
I
long-winded answer, but hopefully it answered the question.
No, there were so many good tidbits of guidance and advice there that it's all awesome. It's a big, good pieces of advice there. Um, if you were to say, like, to give us an example or two of what are those things that you might look back and say, oh, I should have taken that because I didn't know about that. Or I mean, because a lot of us, as you say, it's oh, we do some soul searching. What do I like? What am I like? And so on. But what you're saying is, look at maybe some of the things that you don't like and go after some of those, or things that you see as maybe weaknesses and go after some of those.
Yeah, I mean, so I guess when I said like figure out things you don't like, the way you do that, or at least from my experience, was I just kind of said yes to a lot of different things. Whether or not it was directly applicable or something I thought that was directly going to help me on my CV or on my future applications, it was a lot of just, I'll try it, let's see what happens. And then sometimes you go through that experience, you go, that was awesome, loved it, that's totally what I'm doing now. Or you have the opposite of, I never want to do that again. And I mean, I'm trying to think of a really good example, and I can't think of one off the top of my head. But it was a lot of those. I just said yes to a lot of things. I mean, looking back, there was a lot of classes that it's like hindsight now that I know I want to do computational work and do a lot more of the stuff that I'm doing now. I wish I would have taken different classes, but yeah, it's hard to say when you're first starting your grad school of like, this is where I'm going to go and this is what I'm going to do. I
Sure.
think even taking the time to just sit there, I think we're in a very busy world, right? And you always have to be doing something. Sometimes sitting and doing nothing and just thinking,
Yep.
as scary as being alone with your own thoughts can be, having that moment and sitting there and just trying to actually meaningfully reflect on, okay, what do I like? What did I do? Or what kind of, where do I want to go in this research world? It can be really beneficial. That's actually how I came up with my dissertation topic, or part of it, where it started was I was daydreaming one day and it happened to do with flu, and I was sitting in my principles of epidemiology course. So I probably should have been paying attention. But you daydream and it happens. And I was like, well, we never look at like flooding, and like it all had to deal with like global warming and flooding, and then how I incorporated a flu angle to it. And then a couple people I went to were like, these things are not even related at all. But I was too determined and too stubborn probably to go, I think they are. And I went with
Uh,
it. Yeah.
and are they?
Yeah, actually, at least in
Of
Iowa,
course.
with the population that I looked at. So I gotta be the epidemiologist. I have to say, within my population of Iowa, across the 10 years of data that I looked, there was a percentage increase that, because flooding has molds and can impact your respiratory system, and it can have long-term impacts 6 to 8 months later, and that's flu season. So it can actually make you a little bit more susceptible.
Yeah, and also, if your immune system is busy with the fungal infection, here it comes, and you're
Yep.
more prone to the infection, right? So,
Yep.
so interesting. I really love this connection of behavior with the work that you do. I mean, have you also started to look at it, like psychology or like human
I should. So
behavior?
I really should. I mean, my number one passion is actually teaching. So I want to be a professor. And in fact, I'm looking at jobs now and applying for different academic positions. But I think it all stems back from that one biology course I took my sophomore year. That prof really just had a tremendous impact on who I am as a person, as a researcher, and just as a student. And because it was a lot of like, give your students power, let them do stuff. They'll teach you things too, which was really fun. And so I haven't gotten to the full psychology work aspect, but there is parts of psychology that I like because of education. So
Yeah.
yeah.
Eric, if you were to say a few of the words that he imparted with you, I mean, it seems like this course is awesome. Hands-on course. You are ordering the primers. You are designing the primers and pulling your
Yeah.
hair apart and be like, what am I doing here?
Yeah.
A, T, C's, and G's, and I can't get these things straight. What way is it going? What's 3 prime? What's 5 prime? Oh my God. So, like, all of that experience and going through the process must have been just such an eye-opening experience, to have that responsibility too. But he must have said a few things that stuck with you over the years, and pieces of advice that you may want to tell some of the high school students that are venturing into a
Yeah.
research career.
Um, yeah, so I worked with him a lot. I actually have an email I'm about to send him shortly after this too. But oh man, because I became a teaching assistant for that course too in the end. But I think the most powerful thing he said to me was just be truthful with your students. They'll respond. And as the students, as you're listening to this, understand that for the most part, professors want you to succeed. They want you to do well. But not everybody can be 100% grade A all the time. But if you can trust that that's what they want to do, and then give them the feedback they need to, because they're not perfect, nobody is, But he just said, I distinctly remember the first day of that class, he comes in, he tells all 100 plus students, so this is a 5-credit course. So it was a big class as far as time goes and all that. And he goes, 5 credits isn't enough. This really should probably be a 6 or 7 credit class. You were going to work your butt off, but if you really take it to heart and if you really try your damnedest, you'll do pretty darn well. But you got to give it your all. And what you're going to put into it is what you'll get out of it. I don't think I've ever had a class that exemplified that the way that class did, because it really was. If I poured myself into that class, and it was, he would make a lot of jokes of like, you know, because we're learning photosynthesis and chlorophyll, and he goes, all right, now when everybody goes to lunch, because it was of course right before lunch, and you're eating your salad, just think about all those chlorophyll screaming. And also like he had ways to like just make stuff stick with you once you left the classroom. But it really was, it was a lot of just pour yourself into it and you'll get something out of it. And I like that one class, because there's other parts of it too, that like there was a verbal final that felt a lot like a comprehensive exam. When I went to my PhD, I was like, oh, I've done this before and stuff like that. So there's a lot of other aspects of the course that really prepared me well for just the life I wanted to go on. But it's, if you give yourself to the study, for the most part, you'll figure and you'll really click. And if it doesn't click, don't be afraid to just say, hey, I don't get it. And that was the other big thing that he said, like, it's like, if you don't understand something, just throw your hand up and say, hey, prof guy, what is going on? Because this doesn't make sense to me. And
Yeah.
he would try to find a new way to explain it or have somebody else explain it. So
Yeah. I think that's so important to have the courage and to give yourself the permission to say, I don't know what's going on. Can somebody help me? We don't give ourselves permission to do that, and I don't know why that is. Maybe it comes from our parents, maybe it comes from some of the teachers, or some of our own, our own determination to, perform at our best all the time, and not giving ourselves that opportunity to say, no, but here I have no idea what it is, or I think I know this, but I don't know that. And to be human and say, no, we're all like that. It's okay. This is why we go to school. And I think you put it. I mean, what you learn is you learn how to learn. You don't learn
yeah.
anything.
And to build off that a little bit more too, it's if you're sitting in a class or whatever, anywhere, and you go, I don't think I understand what's happening, or I can't follow, don't be afraid to be that one person that raises their hand, because I can guarantee you there is another large chunk of that student body that also has no clue what's happening. Because I have been that person that sat there lost, bewildered, and didn't say anything. And I wish I would have, because then like talking with my friends after class, they're like, I had no clue. And I was like, oh, I thought I was the only dumb one. They're like, no, we're all dumb together. And I think that's a really important thing of we're all dumb together. So just raise your question and ask, or raise your hand and ask the question, because it only helps. It really does.
Thank you. Thank you for that, Eric. Dr. Eric Kontowicz, so glad we were able to catch up with you and get a little bit of your research and a little bit of your story behind the research and how you got here. Thank you for doing this, and I thank everybody else who's watching this video and for joining us for this Purdue Lecture Hall series. So thanks again, Eric.
Thank you. This was absolutely tremendous. I appreciate it.
Awesome. Well, have a good one. Bye-bye.